of research is focused on human viruses. And in 2013, Dr. Shahid was appointed as a CEO of the DPT/Wellcome Trust Indian Alliance, a biomedical research charity based in India. So without any further ado, would like to again thank Dr. Shahid and welcome him. And the floor is yours, Dr. Shahid, to start your presentation. I would like just to remind everybody here, if you could kindly mute your mic, and if you have any questions, please post in the Q&A tab or leave it on the chat and we will take care of it. Thank you and enjoy the webinar.
Thank you, Dr. Saleh. Assalamu alaikum. Very pleased to be here, and I'm grateful to the American Chemical Society's Kingdom of Saudi Arabia chapter for the invitation, and to my friend Dr. Nahid Siddiqui. Let me give you an overview of this pandemic, and I will in the end hopefully have enough time to seek questions from the audience. So this is the situation of the pandemic as of 8 p.m. today. We have roughly 4.75 million people who are confirmed to be infected in 188 countries around the world. And you will see that over 315,000 people have died as a result of this viral infection. This map shows you where all the virus has been found, where the virus is circulating. And most importantly, you will see these yellow graphs. The first graph here shows the increase in viral infection around the world on a linear scale. And you will see that somewhere around March, the infection really took off around the world. Earlier, it was circulating mainly in China, but in early March, the infection went really all over the world. Interestingly, what you see here is a log scale of the same data. And the log scale shows you something very interesting. The first part of the graph shows a steep curve. This is when the outbreak was happening in China. Then you see a flattening of the curve, and that is when Chinese cases started coming down. But again, the graph took off, and that is when cases moved out of China into rest of the world, primarily in United States and in Western Europe. What you see below that, this graph is the daily number of cases. Sorry, is there a problem with the sharing?
Yes, we cannot see the slides. This first slide is only shown here. Can we show the slideshow?
If you can, please reshare it.
Yeah.
All right. All right. Can you see my first slide?
We cannot see Dr. Shahid.
You cannot. Okay. All right.
It's the green button you will find at the bottom of your screen.
I know. I think it's launching again. I think the problem is with Zoom. It's launching again. Can you see it now?
Yes.
Yes, yes.
Yes. Okay.
But maybe you need to adjust the view to make it full screen, Dr. Shahid.
Yes, I'm doing that. Can you see now, full screen?
Yes. Can you go to next slide? We will see how it is. Next slide.
Can you see next slide?
No.
No, we
It
cannot.
doesn't move.
I'm already on the next slide. I can see it on my end. You see the title slide?
Only
Yes, we
title-
still see the title slide.
Next
Nahmad,
slide.
this is there, my friend.
I think
Can
it's
you
better
have
if you, yeah, if you can, just exit the full screen mode and continue as it is. The full screen
All
mode
right.
is somehow, glitching.
Yeah, I think it's the full screen is problem.
Okay.
Can you see now?
Yes,
Yes.
yes.
Yeah, yeah. Yes.
Okay, wonderful. All right. So let me start over again. This is the condition of the pandemic as of today around 8 p.m., where all over the world we have about 4.75 million confirmed cases in 188 countries. And the virus has so far killed more than 315,000 people. If you look at the graph here, the yellow line, this shows a linear scale increase in the number of cases around the world, cumulative cases. And you will see that the graph really takes off somewhere around early March. Till then, the outbreak was limited to China, but then around early March, it moved out of China. If you look at this graph, it essentially shows you the same thing, except that it plots it on a logarithmic scale, which shows you the early part when the number of cases were going up in China. You have a very steep curve. As China started coming down, the graph started flattening out. But then as cases moved out to the rest of the world, primarily USA and Western Europe, the numbers took off again. And although you can see the graph leveling off a bit, the outbreak is by no means over. And this is very clear from the graph at the bottom, which shows you the daily cases that are coming in. And again, you will see that the daily cases increase from about early March and then have just increased exponentially to have now reached an equilibrium where we are getting roughly 100,000 cases, 80,000 to 100,000 cases on a daily basis. So that's the equilibrium at this time. We will only consider the outbreak to be declining when the daily number of cases start reducing. So that's the fact about the outbreak at this time. I also looked at the situation in Saudi Arabia. And as of today, you have 57,345 cases with 320 deaths. This is roughly 1 point something, less than 2% fatality rate, which is very good because the world over, the average fatality rate is about 7% right now. If you look at this graph here, it shows that the number of people who are recovering from infection, shown by the green line, is roughly 99%. And people who are succumbing to this infection is this orange line. This is roughly 1%, a little less than 1%. So it's a very good situation in Saudi Arabia right now with about 99% recovery. And, for a country of your size, the number of cases is a bit of a worry at 57,000, but the healthcare system is dealing with it quite effectively. Again, plotted on a logarithmic scale, you see the number of cases, cumulative cases. It started off with a sharp curve, and then it has started flattening out, but the curve is still growing. It's by no means coming down. So you still have a growing outbreak, although the outbreak has sort of stabilized, but it is still growing. And you can see this in the daily number of cases. Again, you are getting roughly 800 to 1,000 cases on a daily basis. And this, as you can see, is increasing. So this can be something to worry about. If you look at the deaths, again plotted on a logarithmic scale, follows very closely the number of cases, and the daily deaths are roughly 10 people are dying on a daily basis. And this also remains constant for almost a whole month. So unless these two figures start coming down, you are still not out of waters. Okay. So the question is, where did this virus come from? Well, to understand where this virus came from, first we have to see what this virus is closely related to. And sequences that became available in early January, sequences released by China, showed very quickly that there are two kinds of viruses to which this virus was very closely associated. One group is a group of beta coronaviruses to which the SARS coronavirus from the 2002-2003 outbreak belonged. And the second was the MERS coronavirus, the Middle East Respiratory Syndrome virus, which emerged in your region actually in 2012. The sequence homology to the first SARS was about 80%. The sequence homology to MERS is roughly 50-55%. But surprisingly, this virus showed very high sequence identity to another virus which was discovered in China in 2018. And that virus came from bats. That virus didn't come from humans, it came from bats. And there the sequence homology was 96%. So people immediately thought this virus must have come from a bat. And as you will see on this picture here, bats have been responsible for the SARS-1 as well as MERS. But in both cases, bats did not transmit directly to the human, but it went through an intermediate animal host. So the question was, is the bat virus transmitting directly to humans, or is it coming through an animal source? And now that animal source has been identified, it's a mammal called pangolin. Virus has been found in pangolin, which is roughly 90% similar to the human virus. So the question is, how did this virus come into humans? Was this virus adapted for humans before it entered humans? Or there was a virus that jumped from bats or pangolins into humans and adapted itself? And this is becoming more and more clear now as more and more sequences are becoming available. And this I will come to very quickly. So let us look at the sequences that are now available. And this also really tells you about the power of technology today. Remember, the first sequence of this virus became available in the first week of January. And within 4 months, we have 27,000 genomic sequences that are already in a public database. These are being modeled on a daily basis and displayed at a site called nexttrain.com. And I'm very pleased to see that there are 102 sequences also from the Kingdom of Saudi Arabia in this database. So what does it tell us? Well, first of all, for the virus in Saudi Arabia, this graph here is a phylogenetic tree. It's like a family tree that tells you where the viruses that are circulating in Saudi Arabia really came from. And you will see this, the virus in Saudi Arabia is represented by these blue dots. So there are a few things that you should note here. First of all, these blue dots are distributed all over the tree, which means that the virus in Saudi Arabia came from multiple sources. It is occupying different branches of this tree. So here is really the Western European US branch, and most of your strains, most of your isolates seem to be coming from there. And it is plotted here on a world map also. You will see that this Saudi Arabia is getting virus from US, either directly or through Brazil. It is also getting virus from China. It's getting virus from India, is getting virus from Australia. So really, the whole world is really connected by travel, and therefore it is connected in terms of how viruses circulate. So most of your virus isolates are there, but you see viruses in different branches of this tree that are now circulating in Saudi Arabia. Now, one important point here is the tree is rooted here. We don't know anything before December 2019. And this is really a key to finding where this virus actually came from. So there are two hypotheses. One is that this virus was circulating in bats, and from bats, it came directly into humans. The other is it went from bats to pangolins and to humans. So right now, the hypothesis which is favored is the second one, that it went from bats to pangolins to humans. But as we see, the spike protein, the surface protein of this virus, is very highly adapted to transmission in humans. And this is not the case with either the bat or the pangolin virus. So it appears that this virus at some point came into humans, circulated at a low level, started adapting to humans, started adapting its spike protein based on the human receptor template, and then the infection suddenly took off sometime in December. So it is a key to finding what came before December 2019. And that's why if there are samples available, let's say in China, of pneumonia patients, of influenza-like illness patients in October, in November, we may be able to see the progenitors of this virus in those populations, and then may be able to say very clearly where this virus came from. But that is the situation today, and the world being so interconnected, the virus is really going from one place to the other very easily. All right. A bit about the infectivity and the lethality of this virus. So infectivity and lethality are two different things. Infectivity means how well the virus transmits between one person and another person. And this is defined by something called R0, which means that how many people can one infected person infect. So the R0 for this virus is calculated to be roughly 2.5 to 3, which means that the virus can transmit, one infected person can give the virus to another 2 to 3 persons. And that's the growing outbreak. Typically, you want the R0 to come below 1 so that the outbreak shows a decline. And in many parts of the world, the R0 for this virus remains above 1. It does in Saudi Arabia, it does in India. In China, it has gone down. And unless this comes down because of quarantine, isolation, lockdowns, we're not going to get rid of the outbreak. Okay. The second thing is lethality. How many people does it kill? People that it infects, how many people does it kill? So if you see on this chart, the novel coronavirus is somewhere around here. It's far less transmissible than something like chickenpox or measles, but it is far more lethal than these two. much more lethal than common cold or polio, but far less lethal than something like MERS or even SARS or bird flu. So it's somewhere in the middle. At this time, as I said earlier, the case fatality ratio is about 7%, which means that 7% of people who have been tested positive are succumbing to infection. But that's not the real population mortality rate. The population mortality rate of the virus is likely to be in the range of about 0.5 to 0.7. That's the population mortality. And I say this because roughly for every one person who gets tested positive, there are possibly 4 others who show mild symptoms, who show no symptoms, and therefore they never show up for testing. That's the pattern around the world. The second thing that people have been saying is that, oh, you don't have to worry about this virus. It's just like a flu virus. I'm sorry, it's not like a flu virus. It's far more lethal than a flu virus. And here is the data to show. This shows mortality by common flu. This shows mortality by COVID. And as you see, with increasing age, the mortality in COVID increases, goes up to about 15 to 18% in people who are 80 plus years of age. So the key to reducing lethality is to protect older people. As we get older, we also accumulate other morbidities. Many of us have weak lungs. Some of us have diabetes. Others have heart disease. So all of these are risk factors. And people have seen that there are various clinical manifestations of this disease around the world. So this is not a flu virus. It is more lethal than a flu virus. Flu virus kills less than 0.1%, around 0.1% in the population. This virus kills somewhere around 0.5 to 0.6% in the population. So it's at least 5 to 10 times more lethal than your common flu. Okay. All right, let me tell you a little bit about the virus. The picture on the left is an illustration of the virus surface. This is what we call an enveloped virus. An enveloped virus is a virus that has a core surrounded by a lipid membrane. And because it's a lipid membrane, these viruses are very susceptible to detergents. And that is why hand washing with soap is so important to protect ourselves from this virus. Embedded in the membrane are 3 proteins. One is called the spike protein shown in red. The other is called the envelope protein shown in yellow. And the third is the membrane protein shown here in orange. Now, if you start looking at the architecture of this virus over here, inside there is a genome. The genome is RNA. It is packaged very tightly using a protein called the nucleocapsid protein. And this forms the core of the virus, which is then surrounded by a lipid envelope, which has different membrane proteins embedded in it. It is estimated that the diameter of this virus is about 100 nanometers. And 1 ml of sputum can contain 10 million virus particles. And that's why sputum or fomites are so infectious. And the way you really get infected is if an infected person deposits these either directly when they sneeze or when they speak very close to you, or when they deposit the virus through their secretions on surfaces and you pick it up from surfaces and then you touch your mouth, your nose, your eyes. That's how the virus transmits. All right, so the genome, as I said earlier, shows very high homology to two viruses. One is a bat virus and the other is a pangolin virus, and shows less homology to the first SARS virus, the MERS virus. And there are other coronaviruses that also spread common cold, and this virus is nowhere similar to those. It's a RNA genome, roughly 30 kilobases in length, and it codes for something like 24 to 27 different proteins. RNA viruses traditionally mutate quite rapidly because during replication, there are errors introduced, and RNA viruses don't have an error correcting mechanism, except for coronaviruses. They have error correcting mechanisms. So coronaviruses don't evolve as quickly as, let's say, the flu virus. The evolution rate of these viruses is about 100 to 1,000 times slower than that of a flu virus. If you look at the replication timescales, the virus stays for about 10 minutes on the surface of cells, and then it enters cells. Once it enters cells, it takes about 10 hours to replicate within a given cell. And at the end of this replicative cycle, every virus particle that has entered a cell produces about 1,000 new virus particles. So that is how it spreads. Some people have advocated that if you take hot water and gargle with it, you will not get infected. Well, that has not been scientifically proven, because the virus resides outside cells only for about 10 minutes. So unless you time it very, very appropriately, gargling or drinking hot liquids is not going to do anything, because the virus is already gone inside the cell. So the best protection is a face mask that will protect you from getting the virus in the first place. All right. How does the virus replicate? So to replicate, the virus first has to get inside the cell. So it binds to a molecule called ACE2 on the surface of cells. That's the receptor for this virus. And this binding happens through the spike protein on the surface of the virus. And especially for this virus, it's been seen that the spike protein of this virus is very highly adapted to the human ACE2 protein. But it can also bind inefficiently to the ACE protein coming from some animals like cats, dogs, ferrets, guinea pigs, and many of these transmission has been shown. In fact, you may have heard that one tiger got infected, some cats have been found to be infected, but this infection is not very efficient at all. It circulates best among humans. All right, so once this binding happens, then there is a protease on the surface of cells which cleaves the spike, and this cleavage is a trigger for the viral membrane to fuse with the cell membrane. Once that happens, the virus comes into an endosome, and this endosome is then fused. The viral membrane fuses with the endosome membrane to release the RNA. So here, this step can be inhibited by neutralizing antibodies. If there are antibodies that can bind to this spike protein that prevent its binding to ACE2, then that can prevent virus entry. Secondly, this step is inhibited by hydroxychloroquine, because this step requires acidification of the endosomes, and hydroxychloroquine has a property to inhibit this acidification. Entry of protons into the endosome is inhibited. And that's why some people feel that hydroxychloroquine may work as a drug. Once this RNA is released, the RNA produces proteins, and one of those proteins is a replicase which replicates the RNA molecule to produce more RNA molecules. And it is this step which is inhibited by replicase inhibitors. And you may have heard of drugs like remdesivir, which was developed initially for Ebola, favipiravir, which is an influenza drug. These are being tried in human clinical trials as of now, and they have shown some efficacy. Finally, the fourth step is assembly of the virus particle with proteins that are produced and the RNA. The proteins have to be processed, and this requires a protease to process them. And therefore, protease inhibitors such as ritonavir and lopinavir have also been shown to show some limited efficacy. Again, these drugs were not developed for the coronavirus. These are protease inhibitors that were developed for HIV, and they seem to work to a limited extent in this case. So as of now, there is no specific drug for this virus. However, there are many other drugs that people are trying to see if they would work. Besides acting directly on the virus, there are also drugs in trial that work on the host response, the human response to the virus. That's very important because most of the death happens because of something called a cytokine storm. And if you can prevent cytokine storm, you can prevent death. So what is cytokine storm? And how do you develop pneumonia? Initially, this virus infects the upper respiratory cavity, nasal passages, and throat. It first establishes infection there, and once enough viral particles are formed, then they travel down your trachea into the lungs. And in the lungs, they go to the very tip of the lung and infect these cells that are called alveoli. Alveoli are air sacs, as shown here, where oxygen exchange takes place from the blood into the lungs, and back and forth, CO2 and oxygen exchange. And the virus infects alveoli, destroys alveoli, and you also have a human response, immune response, that tries to kill the virus in the lung. And as a result of this, there is inflammation in the lungs and accumulation of fluid in the alveoli, which compromises air exchange. And this is why people show hypoxia, lack of oxygen, when they get this kind of pneumonia. So again, it's the host response, which is also partly responsible for the situation. Now, let me talk first about drugs, and then I'll go to testing. So at this time, there are multiple drug trials happening. I'm showing you a list here of things that are in phase 3 and late phase 3, phase 4 trials. Remdesivir, which is a replicase inhibitor, hydroxychloroquine, favipiravir, which is an influenza drug, lopinavir, ritonavir, which are protease inhibitors, and let's say a human monoclonal antibody to a molecule called interleukin-6, which is responsible for the cytokine storm. So this antibody dampens down the immune response and therefore hopes to prevent death. Again, this is also an antibody for the same purpose. So many antibodies are being tried and many drugs are being tried. There are essentially two very large trials happening around the world. One is called the WHO's Solidarity Trial, which is testing 4 different things in the trial. And the second trial is something called the Accelerator Trial that is being funded by the Wellcome Trust, Gates Foundation, and MasterCard. And hopefully in a few weeks, we will know what works and what doesn't work. You may have read about remdesivir. NIH was conducting a trial and they opened the trial early, saying that it seems to be working. And now remdesivir appears to be a prescription drug. The only problem with remdesivir is that being a replicase inhibitor, a replication inhibitor, it has to be used fairly early. However, all the therapeutic use of remdesivir is now happening in people who have very bad disease, who have progressed to very bad disease. And that is where this molecule shows very poor efficacy. Yeah, it is like you may have heard of the drug called uh, the drug that was used for influenza for swine flu. I'm forgetting the name. It was useful only when it was given within the first 48 hours of disease. That was a replicase inhibitor. Uh, Oseltamivir, yeah, Tamiflu or oseltamivir. Similarly, remdesivir as a drug should be given early. The trouble is that unlike Tamiflu, which was a tablet, remdesivir has to be given intravenously. So it cannot really be given at home or, to somebody who has mild infection. So it would really be very, very interesting if somebody formulates remdesivir into a tablet, into an oral uh, tablet rather than going for intravenous. Okay. So as you see, much is happening in the world on drug trials. Let me quickly look at testing. Now, there are two kinds of tests available right now. One is the confirmatory test, which is the PCR test. And the PCR test looks for the viral genome, the viral RNA. The second kind of test is called an antibody test. And the antibody test looks for the presence of antibodies to the virus. And both these tests have different values. The PCR test is the confirmatory test. As you will see, PCR becomes positive even before symptoms appear, whereas antibodies become positive only after symptoms. So if you have somebody who is coming with fever on day one, you will not find antibodies, but you will find PCR positive. And the dangerous thing about this virus is that it's PCR positive even before symptoms. So people may be infected, they don't know they are infected, and they are transmitting this virus. This is again a feature of this virus which is very different from the SARS-1 virus. The SARS-1 virus symptoms and PCR positivity were at the same time. Here, PCR positivity is before symptoms appear. That's why this makes it such an efficiently transmitting virus. All right, so PCR is the confirmatory test. Antibody test is a more of a population-based test to say how many people got infected. The rapid test that is being circulating looks something like this. And this can give you 3 lines. And I've shown you the scenario here. This situation is a negative. It has only 1 line, which means the test worked, but nothing else. This line means that it is IgM positive. This line means it's IgG positive. And if you have both lines, it means that you have both IgG and IgM. So what do these really mean? Well, this means negative, no antibody, but it doesn't mean that one doesn't have the virus. You may be in the early phase of infection. This test means that you are in early phase of infection. Both positive mean that it's a recent infection. Only IgG positive means it's a past infection. So it's important to understand this test clearly as to what it can do and what it cannot do. Recently, some ELISA tests are also getting approved. And the difference between the rapid test and the ELISA test is that the rapid test is only qualitative. It will tell you whether infection is there or not. Whereas the ELISA test also tells you the level of antibodies, the quantity of antibodies, and that becomes important when you talk about vaccines and protection and all that. All right. Go on through this. Finally, coming to vaccines, and it's really vaccines which has shown the power of technology today. Uh, within 4 months of the sequence of this virus being known, we have over 130 vaccine candidates that are being developed. About a dozen of these are in fairly late stage of development, and 6 of these have already gone into human trials. The first vaccine that went into human trials is an mRNA vaccine by a US company called Moderna. This vaccine, from the time the sequence became available to the time this went into humans, was 63 days, 2 months, which is phenomenal. It has never happened anywhere, any time earlier. The closest we did was 20 months at the time of SARS-1. So this really is the hope. This is where technology is going. And companies are moving very rapidly to develop these drugs. So for example, there is this vaccine here, the Chadox vaccine, which is being developed at Oxford University. It's also in human trial. There is an Indian company called Serum Institute of India. They have already started manufacturing this virus even before the trials are over. They are hoping that it will be successful, and then they will have a vaccine ready. This has never happened. So what COVID is also doing is completely changing the paradigm of drug development, drug testing, vaccine development, vaccine testing. Many times animal trials and human trials are going in parallelly. Earlier paradigms was you first test it in animals. If it's successful, then you go to humans. COVID has completely changed all of that. This figure here essentially shows you the different kinds of vaccines that are being developed. These may be live attenuated, inactivated vaccines, viral vectors, either non-replicating or replicating, recombinant proteins, peptides, virus-like particles, DNA, RNA, all kinds of vaccines are being developed. So this is fantastic. This is really where technology is today. All right, so I'm pretty much done. I wanted to put this out because our organization for the last 2 months has been doing a lot of public service, a lot of public engagement work, and we have been putting out these infographics on COVID. We've essentially put out 4 infographics, 1, 2, 3, 4, and all of these infographics we have also translated into about a dozen Indian languages. If there is any interest from Saudi Arabia, we'll be happy to send you the template so that somebody can translate this into Arabic, and you are free to circulate it as you wish. You can go to the Coronavirus Resource Hub on our website, where you'll get a lot of other information for this virus. And finally, let me just finish with some key messages. Well, first of all, I think it's very important for all of us to follow local government guidelines and WHO guidelines. These are experts who are telling you what should be done, what should not be done. Second is, we don't really know how this virus transmits, how it will affect each one of us. So each one of us needs to consider ourselves to be infected, and it is our duty to protect everyone around us. And the only way we can do this is to wear masks when you are in public and frequently wash hands with soap and water. If that is not accessible, use hand sanitizers frequently, but use sanitizers only that contain at least 68 to 70% alcohol in it. There are many myths that are floating around. I've listed a few of these. One myth is that there are strong and weak strains of the virus circulating in the world. That is completely untrue. As I showed you in the family tree, there are multiple isolates floating around, but nobody has found whether one isolate causes bad disease versus another isolate. So don't be under this impression if somebody tells you that in your area or your country, it's a weak virus and you don't have to do anything. It's completely wrong. The second is that chloroquine or hydroxychloroquine can be taken as a preventive. It should not be taken by people just to prevent getting infected. It won't work. It will actually cause you far more harm than it will cause good. The third myth that has been floating around is that people who have had malaria or who have BCG vaccination are protected against COVID. There are studies coming out now that are showing this is not true at all. This correlation doesn't really hold up. And the fourth one on my list is people telling you that summer temperatures will end the pandemic. There is no evidence for it that summer temperatures will end the pandemic. The virus is transmitting from one human to another. It is finding very little time outside in the atmosphere. And that's why we don't really think that temperatures will do anything to it. Don't wait for a drug or vaccine. A vaccine is at least 12 months away. A drug, we don't really have a drug fully developed for this virus. It is up to us to break the transmission and to protect ourselves. Lockdowns, isolations, quarantines are difficult, and they are extremely difficult on poor people, as we have been seeing the world over. So please, this is the month of Ramadan. Help those who are less fortunate. Those we can help, we must help. I will end there and take any questions. Thank you very much.
Thank you, Dr. Shahid, for this informative presentation and educative at the same time. Yes, we do have some questions already posted in the QA tab. I don't know whether you would be able to look at them and answer them. And also we have the people who would like to raise hand and give a live questions to you as well.
Yeah, so Abdul Hafiz, well, Abdul Hafiz, Assalamu alaikum. I do remember you from ICGB. He is asking, since this virus is mutating quite slowly as compared to flu virus, people who are infected once, are they protected for subsequent infections? That's his first question. Let me answer that first. Most likely people who are infected once will be protected for some time, but what we don't know is how long neutralizing antibodies last. Those studies haven't really been done. So going by other coronaviruses that have become endemic, antibodies do last for less than a year, protective antibodies, because you get common cold by those viruses every year. SARS, the studies that have been done again show that neutralizing antibodies possibly last a year. So most likely neutralizing antibodies to this virus are not going to be very long lasting. It is going to become endemic in the population, and it remains to be seen whether if you get infected once, whether you can be infected the next year or the year after. We just don't know that. We haven't been there yet. The second is, will herd immunity bring down transmission eventually? Eventually, yes. Eventually, all viruses, you get protection through herd immunity. And there are two ways to develop herd immunity. One is once enough people get infected and develop antibodies to break transmission, you get herd immunity. And for that, roughly 60% of people have to be 60% of people have to be infected. The second is through vaccination. Again, if you can get a vaccine made quickly and you can protect about 60 to 70% people around the world, that's another way to develop herd immunity. But herd immunity, developing it Sweden style, is extremely dangerous for countries like India, where if we started doing that, there would be something like 4 million to 5 million deaths before we get to herd immunity. Okay, so the second case is by Mohammed Ansari. Thank you very much for this informative talk. I'm curious to know if remdesivir may be late and may not be successful. Yes, so that is exactly, Mohammed Ansari, that's exactly what I was talking about. Remdesivir is a replicase inhibitor, and replicase inhibitors have to be given early to break the potential for transmission. Just like Tamiflu doesn't work after 48 hours. My suspicion is that remdesivir is also not going to work very well if it is given late in disease. But the trouble is that remdesivir has to be given intravenously. So the key is whether somebody can make an oral remdesivir. Danish, Moderna's early trial of coronavirus vaccine produced antibodies, right? That's true. Phase 1, they're now going into phase 2. Produced antibodies, the key thing is whether those antibodies are protective or not, whether they're neutralizing antibodies or not. And they will do what are known as neutralization assays before we understand that. Afsar Azmi, majority of patients come with acute symptoms in the hospital. A major proportion of patients on ventilators do survive. Before an effective vaccine is developed, what is your opinion or suggestion to help doctors in the management to reduce mortality? Well, I'm not a doctor, so I don't really want to give advice on how to really save patients. But from what I've been reading, try not to get people on ventilator too quickly. It's better to manage oxygen and ventilation through BiPAP rather than a ventilator. And this is something that many of my doctor friends have been saying, that once somebody goes on a ventilator, it becomes very difficult to wean them off. Can you please elaborate if the virus can enter through the eyes? Yes, absolutely. Many viruses enter through the eyes. And you have to remember that eye is an immune privileged organ. There are no immune cells, there are no antibodies in eye. So many viruses, including flu viruses, enter through the eye. So yes, don't rub eyes with infected hands. So that's why, 3 sites are very important: mouth, nose, and eyes. Try not to, have dirty hands on those. What could be the reason behind more deaths in USA compared to Saudi Arabia? Well, I mean, I think USA ignored advice and didn't take things seriously, and the virus really took off. That's precisely it. And if you see everything that's happening there, the political leadership is not listening to the doctors and scientists, and they continue to pay a price for it.
Abu Muhammad, can we ask a question rather reading?
Yes.
I suggest that let Dr. Shah to complete the written questions, and then we would
Yeah,
go
for
for
many
the
written
so,
ones.
Ashraf Al-Tahini, interesting question. There is a conspiracy that this virus is human-made and not nature-created illness. So what are your thoughts? Well, my thoughts are the same as that of anyone who follows science. This virus is not created in a lab. This virus could not be created in a lab. And I'll tell you the reason. If you wanted to create a virus in a lab, you would use a backbone from a virus that was known to cause human disease. This virus does not have a backbone from any virus that was known to cause human disease. Its closest relative is a bat virus that was never found to cause human disease. So logically, nobody would use a virus not known to cause human disease to engineer a virus. If somebody wanted to engineer this virus, they would have taken the SARS virus and made it more efficient. But SARS virus has only 80% homology to this, 96% homology to a bat virus. And also, if you look at the spike protein and the receptor binding domain in the spike protein, it clearly has evolved. If you put it through a computer prediction, computer would not have predicted those changes which the virus has made. So it's clearly not something made in a lab. You know, it's interesting when you create something in a lab, the changes are very abrupt. When this happens through evolution, the changes are slow, and we are seeing signs of slow evolution. So I don't believe that it was created in a lab anywhere. So somebody's asking whether taking hot water vapor through the nose can kill the virus. Only if the virus is outside the cell, which is about 10 minutes. So you have to be doing this inhalation pretty frequently during the day. Don't just bank on it. Steam inhalation, gargling, all those are good hygienic practices. Keep following them. But if you think that it's going to kill the virus and keep it protected, I have my doubts. Is it true that the virus is aggressive on certain genes? I don't really understand what Dr. Rohama Gill means by this. Maybe you can clarify this. How effective is the use of surgical masks by public? Please don't wear a surgical mask, wear a cloth mask. That's very effective. Leave the surgical mask to surgeons. This is Server from Michigan. Is it airborne? And if so, how much chance of getting infection out there jogging? If you're wearing a mask, you're all right. No diagnosis is possible in asymptomatic person. This is correct. Also, Dr. Rohama is asking gene of the host tissue. So yes, the virus seems to be leading to a cytokine storm, which is a host host response, and we really don't fully understand why that's happening. So in a sense, yes, it does activate host processes, and many viruses do. It's not just this virus. Many viruses do. I think those were the questions. If somebody has a question to ask, please feel free.
Yeah, I think we have some questions from the participant here. Suhail, yes. You have one?
Can I ask a question?
Yes, please go ahead.
Yeah. Okay. Dr. Shahid, thank you very much for your nice, excellent presentation. A lot of information we learned from this nice presentation. And also, thank you for you willing to share this infographic information, because we have a plan to use actually some kind of booklet from our chapter to circulate on coronavirus. I think our one of the BOD, Dr. Amani, she's going to contact you, and then
Yes,
please
please,
share.
please send me an email and I will send you an editable copy where
Sure.
you can replace the English with Arabic.
No problem. Thank you. Thank you very much. So my question is very simple question. You know, you show the map of actually how the virus transfer from different country. They have different country, have different kind of genetic information. So in Middle East, it shows mostly is coming from Europe and USA, right? Compared to Asia, the mapping it shows
Yes.
high
Yes.
similarity to US and Europe.
Yes.
Oh, okay. Thank you.
But there are isolates circulating in Saudi Arabia that also show links to viruses found in China, India, Australia.
But majority of the strain coming from Europe and US, yeah?
Majority is Europe and USA. Yeah.
Thank you.
Do we have any more questions?
Yeah, we do have an attendee willing to ask a question, Mr. Saroj.
Please go ahead. Yeah, please unmute yourself and ask.
Saroj, can you hear us? If you could ask your question, please.
He is not around. Muhammad, can I ask my question?
Oh, certainly, Abu Talal, go ahead.
Okay, first, Dr. Shah, thank you very much for this very informative presentation. So beside backbone and human-made virus theories, my question is very philosophical, you know, just why this virus is very selective and attacking heavily very powerful countries like US, Europe, Russia, and very rich countries like GCC, while very poor countries in Africa and Asia look like immune from this virus, in spite of their limited and scarcity of resources. Can you comment on this, please?
Yes. So first of all, the rich countries did not pay attention early enough. And two great examples of that are US and UK. UK only started taking it seriously when Boris Johnson got infected. Unfortunately, Trump hasn't been infected yet, so it's still taking US a little time to understand this problem. You know, in poorer countries, I really don't know whether there isn't enough infection or we're not measuring it. If you look at African countries, for example, the level of testing is really very low. And if you don't test it, you really don't see it. So I have my doubts very much that many of the countries that are showing very low levels, very low number of cases, that is true. I think it's a lot to do with less number of tests. I have seen this in my country. As the number of tests have started going up, and we are now testing in India, we are now testing roughly 40,000 tests a day, PCR tests. And as the number of tests are going up, we find that the number of positives are also going up, despite the lockdown that we have had for about 50 plus days now. So I think it's reflective of testing, really.
Okay, thank you very much.
Hello. Hello.
Yes. Yeah, go ahead, please.
Yeah, this is Saroj. Sorry to bother you. Okay, thank you so much, Dr. Jamil. It is a very interesting presentation, and thanks to organizer. I have two questions. Myself, Saroj, and I have first question. For example, there are two major sources of infection. One is the touching, and second thing is the droplet, if it coming through the nose. For example, what should be the ideal contact time? Like it is in seconds or minutes that will cause you infection, or what is there any data for that?
Well, first let's first look at the aerosol. So aerosol, it's been seen in studies that if somebody is speaking, then the aerosol they are generating usually doesn't go beyond about 3 feet. If somebody sneezed, then that aerosol goes much farther. And you can either take the aerosol directly if you are very close to the person, or that aerosol settles on a surface and another person picks it up from the surface. So here, if you also look at how long the virus can survive on surfaces, it can survive on plastic and steel surfaces for up to 3 days, 72 hours. It can survive on cardboard surfaces for up to 24 hours. So that's why that is really the biggest source of infection. When we touch a doorknob or we rub our hand on a table, we don't know who's been there, and whether it's infected or not. So that's why routine cleaning of surfaces is very important. And more importantly, for individuals to be washing our hands, sanitizing our hands frequently, especially if you're coming from outside, coming inside your homes. The second thing is try not to touch your face with your hands. That's a very easy source of infection. When you pick up from surfaces, put your hand in your mouth or your nose or touch your eyes. Those are the 3 things that you need to protect. And that's why a mask helps. If you have a mask on, you cannot touch your mouth, you cannot touch your nose. And if you have a mask and you have glasses, then you can't even touch your eyes. So that's how it protects. Mask also protects if somebody sneezes very close to you, you will not get the full blast of it. You yourself will not give out virus if you are infected, if you're wearing a mask. So the amount of virus in the environment reduces if everyone starts wearing a mask. So everyone must wear a mask in public. And I think this is going to be the norm, at least for the next 6 months and probably more. People need to be wearing a mask in public.
Dr. Jamil, thank you. I
Yes.
have just a leading question here. Exactly whatever you said, maybe many experts will advise. Now the working place environment industry people, they will accept. This is very easy considering the present situation. But have you considered how long a person should wear the face mask, considering the side effect of wearing face masks, like reduced level of oxygen and all these things I was reading, it is more harmful. So there should be any recommendation, or whenever we are going to workplace, now the company or the institution, they will make a strict rule, put the face mask. But how long it is ideal? 3 hours, 4 hours, 2 hours, or 8 hours? Means it is good to put in the people's face.
So when you are saying that it reduces oxygen, you are essentially talking about an N95 mask.
Yes.
I'm not advocating that you wear an N95 mask. In fact, you should not wear an N95 mask. Leave the N95 mask to healthcare workers. You please wear a simple cloth mask. Make it at home. If you want, I can send you ways to make a mask. That mask you can wear all day. It's no problem. There's good oxygen exchange through it. Those are the masks we are advocating that people should wear, a cloth mask. Don't wear N95, please don't.
Okay, thank you so much. Thank you. That's it.
I think we still have 2 other questions, Abdul Hafid and Muhammad Ansari. If anyone of you would like to ask the question.
I have a third question, Saleh, if you allow me. at the end. Okay.
I see two other questions here. Isha Altaf, why is spread of coronavirus fast as compared to SARS? Very good question. If you remember, I told you that in SARS, the virus became, people became virus positive at the same time as symptoms. So you protected yourself, you isolated yourself. Here you are shedding virus at least 3-4 days before you show symptoms. So you don't even know that you're infected and you're spreading it out. So that is why this virus has gone about. The second thing is that this virus has developed its receptor binding domain in the spike protein has evolved to make a very tight fit with the ACE2 receptor. So it binds to the receptor much better than the SARS spike protein did. So there are essentially these two reasons. One is a biological reason that the virus fits very nicely with the receptor, can transmit very easily, and the second is that you may be shedding virus for a few days before you get symptoms, so you're not careful. And these two reasons. Dr. Sadia, is there any correlation between environment conditions and COVID cases? I think when you say environment, you're probably talking about temperature, you're talking about humidity, you're talking about environmental pollution. Temperature and humidity, people have shown some correlations, but there is really no evidence that the virus transmits better or worse in some, as a result of temperature. But in cold temperatures, people usually stay inside and they stay in closed spaces. So the effect of cold temperature likely is that you stay in confined spaces and give the virus better opportunity to transmit. Environmental pollution, definitely, because it affects your lungs. It affects the lining of your respiratory tract. And this can be a comorbidity for this virus, as for any other respiratory virus. So from that point of view, yes.
Sir, I have a question.
Yes, let me just finish one other question on the list here. How much one should be believing scientific literature coming from China? Well, I mean, I think we are targeting China for all the wrong reasons here. Science is science. I mean, if you are a scientist, evaluate the scientific information. Eventually, if something is wrong, somebody proves it very quickly these days. There are, over the last 3 months, just in the public domain, there are close to 5,000 papers. Obviously, everything is not peer reviewed. So use your scientific judgment, but to blanket, to say that if something is coming from China, I will not trust it. But if something is coming from the US, I will trust it. That's not being very scientific, is it? So I wouldn't advise that. All right, please go ahead, ask your question.
Assalamualaikum, this is Yunus, sir. So my first question is,
Assalam, Yunus.
What is your opinion about virus transmission from dead bodies?
Well, dead bodies would have secretions, and secretions transmit. As we have seen in this case, even stool is positive for a fairly long time actually. So dead bodies will passively transmit from secretions. Of course, dead bodies will not actively transmit. Dead body will not sneeze, for example. So passive transmission, yes, active transmission, no. Have to take all the precautions.
About remdesivir that it may be late. So what is your opinion about use of neutralizing antibodies or people donating plasma? How effective it would be? What do you think?
Yeah, so even plasma would have to be given fairly early. If plasma is given late, I don't think it would make much difference. I mean, it will slow down, of course, it will slow down. But even plasma therapy has come into a lot of controversy simply because people who get bad disease really develop high affinity neutralizing antibodies, whereas people who have only a mild infection don't really develop very high quality antibodies. So somebody who's already very sick, before they leave the hospital, you're trying to bleed them for plasma. That itself is a matter of concern. So in this case, I don't really think plasma therapy is a very viable option. But if we can quickly develop human monoclonal antibodies which are neutralizing, that would be a real advantage. And there are people who are doing this at really very good speed, and some are becoming available at least at the testing level.
Thank you very much. Thank you. Thank you, sir.
I think we have two questions in the Q&A tab, Dr. Shahid.
Yes. Why are asymptomatic cases in India are more compared to other countries? Well, that's not true, really. In India, as well as everywhere else, about 70%, 70 to 80% people either have no symptoms or have very mild symptoms. So it's not true that people in India are more asymptomatic than elsewhere. India also has not been testing to scale, so it may also be a testing issue. And then there is Zulfazal. Hello, Zulfazal. Assalamu alaikum. How are you? How our body immunity will be helpful in protecting us from COVID-19? Well, having a good, strong immune system, as Zulfazal, you know very well, being a virologist and an immunologist yourself, innate immunity restricts many viral infections before they can get very bad. So if you have strong immunity, the first stage, the innate immunity, is likely to control this infection, or at least dampen it to a point where it will not really cause much harm. So yes, do everything to build up your immunity. Eat well, sleep well, all of those.
Is Mr. Ansari still there? I can see his hands up. Ansari, do you still have a question, Muhammad? Or if not, then we'll move to Dr. Nahid Siddiqui. Please go ahead, Dr. Muhammad.
Okay, thank you. Thank you very much, Dr. Shahid. My question is, it is just comparative. I mean, yesterday or day before, we were reading a pharmaceutical company in California, I think San Diego, and they came up with the idea, I think that's Sorrento, if I can pronounce it properly, that they have developed the antibodies which are going to block 100%. So if you take the antibodies in development, which is going to take a long way, or they have the longer route actually. So how do you see these antibodies if developed, if the FDA is going to approve, then it going to be game changer for this COVID control?
Certainly, I think antibodies, if they have developed humanized monoclonals, neutralizing monoclonals, that would be a very good step. I think many of us don't remember that till maybe about 20 years back, even for hepatitis A, there used to be passive immunization. When you were traveling, you would get serum gamma globulin for hepatitis A. So preventive antibodies have been used for a very long time. So I think if a company has developed human monoclonals that are stable enough to remain in the system, they can be used as a preventive. But more importantly, they can be used as a therapeutic. So that would be a very good development.
There's two questions in the QA, Dr. Shahid.
Yes, I can see Zain Qadri, who is a former student of mine. Zain, assalamu alaikum, how are you? Is there any link of COVID to neurodegeneration? Yes, I don't remember the cell, the exact cell that has the ACE2 receptor, but people have shown that there are neurological symptoms also associated with COVID. It has, all kinds of manifestations, including, so I'm not sure whether it's neurodegeneration or it may be something else, but certainly neurological symptoms are associated with COVID. Wajil Khan, what is your opinion about gender-based infection? So I wouldn't say gender-based infection, but certainly the mortality has been shown that the virus is killing more men than women. So mortality is definitely skewed in favor of women. I really don't know what the mechanism is. Dr. Rohama Gill, like other viruses, will the novel coronavirus stay with us? Absolutely. It's not going away for the next few years, and there is all indication that it will become endemic to the human population. Right, I don't see any other questions here.
Well, if there are no other questions, let me just ask them this question, Doctor. So
Sure.
When do you expect life will go normal globally with this coronavirus? Especially that we've seen now some European countries somehow are getting back to normal or kind of ignoring what's going on there.
Well, it depends. It depends what you call normal. If wearing a mask in public is normal while you go about your daily lives, I guess fine. But I think our lives are going to be changed fundamentally from before COVID. We will not make flights that many. We will not fly that much anymore. We will not travel that much anymore. We will do a lot more online meetings than physical meetings. We will be more wary of protecting ourselves in public spaces. I think if you see a lot of companies are moving towards work from home now. So that will become the new normal. So if by normal you mean when will we go back to the way we were living before COVID? I think never. And in a way that will be good, because the way we were living was not sustainable. So we'll see, but at least in the short term, over the next year to 2 years, things are going to be very different. Very, very different.
Well, at the end of this webinar, we would like to, on behalf of the board of directors of the society and its member, would like to extend our appreciation to Dr. Shahid Jamil for this very educative and informative presentation. I would like also to thanks to organizer and every participant in this webinar. Hope to see you in future webinars for the Chemical Society, the Saudi chapter of it. And I think we have one more announcement. Maybe Dr. Bashir, if you'd like to conclude with that announcement about the GCA conference.
Well, before Dr. Bashir, let me just say that, you know, I really enjoyed being here. Thank you for inviting me to give this webinar. My slides, if anyone wants, please just write to me, or maybe I can send it to you, Dr. Nahid, and you can distribute to whoever wants it. Yeah. And if you are interested in the infographics, I will be very happy to share the editable versions. We would be very pleased if they are translated in Arabic and distributed not just in Saudi Arabia, but in the entire Arabic-speaking world. We would be honored if you did that.
We will do that.
Thank you again.
Thank you. Thank you. Thank you, everybody. Actually, we have a conference in November in Bahrain. And we have our first conference in GCA. It's called Gulf Chemistry Association. Call for abstract is still open. And then the end of June is the last day for submission of abstract. So I urge the participants kindly submit your abstracts to the conference. Thank you very much.
I think we should also thank Dr. Hind, because she was communicating with Dr. Shahid, Dr. Bashir.
Thank you for Dr. Hind, yes, and for you and for your nice organization. It's excellent presentation, Professor Shahid. And if you have time, you can join our conference, inshallah, in November. Thank you.
Thank you. Okay, thank you, everybody. Right. Thank you, Dr. Hind.
Thank you very much.
Abu Muhammad, can we have the BODs just after this presentation for 10 minutes?
أخوي بنروح ننام يا أخوي ورانا شغل الحين.
It was a pleasure, and hope to see you sometimes in our other meetings. We are, for the GCA, it's not official, but we are actively, yesterday discussing that we may include one session for the COVID-19. I don't know yet, but one of the BOD proposed. We'll see how it is moving. If there's something development, we will contact you, inshallah.
Well, that's it for the webinar. The POD meeting of Talal, I don't know if we can have another session for it. We still have more than 30 members connected to the webinar. We'll take them a while to leave.
They can leave in 5 minutes. They can leave in 5 minutes, I think. No one can help to leave them.
Thank you, Dr. Siddiqui. Thank you, Mr. Saleh.
It's a pleasure. You worked hard and
distribution. Yes, thank you very much.
Now I will write email to Dr. Shahid and we'll get the information. I think we got the job to translate those in info.
Yes.
And then it will be good, inshallah. It's good.
Yeah.
Where are we standing, Nu'man?
We still have few attendees.
Okay, tell us when we have all the
You can request them.
Sure.
You can request
for sure.
a message. So we can have it here. Noman, did you capture the screen?
Yes, I did.
Oh, great. That's wonderful.
هو around.
Wait.
السلام عليكم.
عليكم السلام.
عليكم السلام.
مساكم الله بالخير جميعا.
الله يسعدك، يعطيك الف عافيه.
الله يعطيكم العافية.
وبركاته. الله يعافيكم.
If we are planning to discuss something about GCA, we should have Jamal and Abu Saleh also.
Yeah, just I have some question to you guys. Do you think that if we publish the presentation of the speaker is a good idea in our website? Or
Yeah,
no?
sure.
Or just give like abstract as the professional website they did it? Because
There is a one session for you for webinar events. You can put all the information.
هلا ويا.
which one? The website you mean?
هلا حبيب قلبي أنا بس عندي
عندنا all information related to webinar.
عندي الحين الويبينار حق
I didn't get what you said.
كورونا مع
All the information regarding webinar you can update.
Okay, I will go to this
So
website.
no one can help you to add this information and your
No,
name.
we already generate one website with all the information.
Yeah.
But I mean to put the presentation for each speaker is not the appropriate way, right?
No, not presentation, the information of
Yes,
webinar.
information. Yes, correct.
Yeah.
Yeah. Okay, yeah, got it.
Also, can we upload screenshots online on website?
Yeah.
As a gallery.
Yeah, why not? And even for that, Noman, I didn't see in the Teams my name there.
BOD?
Yeah.
No, it's there.
No, the member, speaker, sponsorship, membership, event, social program, publicity.
Okay. You're talking about the main menu.
Yes.
Yes, I am still waiting. I was still waiting for your confirmation that if everything is fine.
Yeah, everything is okay.
Fine. All right. I will publish the page now.
Regarding the screenshots of the webinars, we can utilize the social media just to show people how was the interaction, how was our webinars just a few photos we can post them.
Yeah, definitely. I mean, give it to the people those who are dealing with the social media, and also in the gallery, because we had 4 webinars, so their screenshot should be there to show them that how many people are attending, or how important our webinars are, how successful we are conducting.
I just received a message from one of my friends in Canada. He connected to the webinar, and he really appreciated the information that he received. And he took snapshots and passed them in a WhatsApp group for his people. So I think it was very good selection tonight. Thank you for Dr. Hand, Dr. Siddiqui.
Thank you for you guys and for everyone participating in this. I would like just to share the screen with you guys to see. Yeah, did you see this, the homepage of this event?
Yeah, we can see, but it's all black.
And now